Hello, my lovely anatomist and physiologist, Michele here. We are finally ready to move into the stomach, which I think, really for me, before I studied the digestive system, I just assumed like this was the place where digestion was taking place, and that's not actually the case. I have here a sagittal cut of a stomach model, a picture of a sagittal cut of a stomach model, and I want to go ahead and take a look at this to begin. So remember, we left off talking about the esophagus as the tube that's going to be propelling your bolus from the mouth into the stomach. And so we're starting out with this small tube or passageway that's leading to the stomach. And when empty, the stomach is about the size of your fist, but it can enlarge depending on the amount of food that you've ingested. And so the shape of the stomach is maybe like the letter J on the side, maybe we could say. I'm not sure exactly how to describe it. When we are moving from the esophagus into the stomach, remember we have what we described as the esophageal sphincter. And sphincters are circular muscles that are going to be closing off entries and exit points. And so here, this is preventing acid from moving from the stomach up into the esophagus. When acid from the stomach moves into the esophagus, we talk about that as acid reflux. And we'll see that the esophagus doesn't have the protective mechanisms to deal with that acid that the stomach does. And so we can actually see damage being done to the tissue, the lining of the esophagus. Let's also go ahead and mention the stomach. So I'm just going to label the bulk of this organ here, the stomach. Leading from the stomach, you're going to see material moves into the small intestine. And specifically, we're going to go into the duodenum of the small intestine. And so this is that beginning portion of the small intestine. But again, you have another sphincter here in between the stomach and the small intestine. And we do call this one the pyloric sphincter. And so this just gives us a little bit of framework for some of our conversation. Now, remember, there's going to be more structures that you're learning in terms of your anatomy of your stomach, and I'm going to let you pick up that level of detail in your lab section. So you're able to do that already in the lab. So let's go back to thinking about what is the job of the stomach? And it seems like for me, before I studied it, it seemed like the job of the stomach was digestion. And yes, but not so much. Like really, the job of the stomach is to hold your food. Job of the stomach is basically like hold your food. And so I'm trying to look for this information. So the stomach is going to hold, it can hold up to no, let me rephrase. The stomach can hold 4+ liters of food and liquid. My gosh. Think of like a 2 liter of soda, but 2 liters. Woo! And then, remember that's a 4+ liters. So depending on how much material that you're eating. And I think it's fair for us to think about this material as being in the stomach for about 3 to 4 hours, depending again on the amount of food that you're eating, how long it's going to take to actually process that food. Now we do see some digestion is happening here. So we're going to get some mechanical. And we're going to see that's the segmentation type of movement. And we're going to get some chemical digestion happening here as well. So we're going to go into the specifics of that as we go through in just a few minutes. Let's begin by talking about our gastric secretions. And I'm going to put activity. So I'm saying gastric secretions activity regulated by. I'm not sure if that's the best title or heading, but that's the one that we're going to be, that's what we've got. So that's what we're going to use. So we've actually already talked about the very first part of this as the cephalic phase, where cephala is your head. So this is that conscious level, I'm going to say awareness of like food. So here you can see it, you can think about it, you can smell it. So sight, thought, smell, taste. Okay, this is a very rapid, really quick CNS control to increase both your gastric secretions and motility and your saliva. So really getting on board, ready for both chemical and mechanical digestion. The second phase is described as the gastric phase. And this phase can last 3 to 4 hours. Again, and this is depending on how much and the type of food that you've eaten. And so this is triggered by stretch receptors in the actual stomach. So food has distended stomach or stretched the stomach. And okay, just I'm using the term food here very loosely, right? Because, you know, technically we'd call it the bolus if we're down in the stomach. But that seems a little bit unnecessary to say that. And what we're going to see also is the actual, you know, chemoreception of the food. Again, meaning we're going to have differences in whether it's carbohydrate heavy or lipid heavy or protein heavy. And the gastric phase is characterized by an increase oh. Okay, I had something a little bit confusing in my notes. So just to clarify, in the gastric phase, we're going to see if you have the stretch receptor stimulated because the stomach is stretched out, if you have chemoreception of food material that has entered. And I see that's kind of a sloppy writing, so let me try to improve that. So you can do commas for these. And if you have a rise in the pH, if you have a rise in the pH, so the pH is more basic, meaning you haven't been making a lot of hydrochloric acid, then all of these are going to increase the release of a hormone called gastrin. And we're going to be talking more about gastrin here just in a little bit, and we'll talk about it several times. So you might want to highlight, I'm going to circle, I can't get to my highlighter, gastrin. Okay. The third phase is described as the intestinal phase. The intestinal phase. And here what we see is this is triggered by the food entering the duodenum, which is, of course, the top portion of the small intestine. And this will actually slow gastric activity. This is so interesting to me. So the actual stretch of food entering the duodenum, the beginning part of the small intestine, will slow down the activity of the stomach. So here's the thing I always picture, and some of you are going to have to go to YouTube to look this up. I'll see if I can, I'll maybe stick it into our playlist if I can find it for us. But there's a old TV show called I Love Lucy. And in this show, Lucy was always getting into trouble. And so she and her friend Ethel are in this chocolate factory, and it's their job to package the chocolate into the box. And the machine speeds up and speeds up and speeds up. And pretty soon it gets to the point where Lucy and Ethel can't keep up with the chocolates. And so they're shoving the chocolates in their mouth, and they're shoving the chocolates down their dress, and they're shoving the chocolates into the pockets, and all this, is going like everywhere but in the boxes. And it's just a total disaster. And so this is what we're trying to avoid in our digestive system. So essentially, we have these small, I like to think of squirts. So we have these small little squirts of chyme from the stomach going into the small intestine. And then the small intestine has to tell the stomach, hey, whoa, stop. Like, I need time to process this material. I need to fully digest and fully absorb the nutrients from this chyme I have right here. So a little bit goes into the small intestine, and then we tell the stomach to stop, and we process what we have. And then a little bit more enters into the small intestine, and then we tell the stomach to stop, and then we process it. And then that continues. And this is why it's taking like 3 to 4 hours to digest a meal, because you're doing it just these tiny little, I'm going to say, squirts at a time. I actually have a number that I wrote down, and I think it is like 3 milliliters. It's 3 milliliters. It's such a tiny amount, it's such a tiny amount of liquid. And so that's what we're doing, a little 3 milliliter squirt, and then stop, let me process this. So 3 little milliliter squirt, and then stop, let me process that. So that's what's going on with that intestinal phase. And we can talk about this as an Anteroendocrine Reflex, where the entero here is talking about enteric, the gut, the GI tract. Okay, so I'm gonna square that, because again, I can't get to my highlighter. Okay. Okay, got my system up and working. So now I've highlighted my gastrin, and I've highlighted my enteroendocrine reflex. Okay, taking a look, conversation about the stomach. We're going to be picking out 3 different types of cells that are lining the stomach, and talking about what kind of chemicals they're producing. And so let's list out our 3 cell types. So we're going to have the parietal cell, we're going to have the chief cell, and these are cells that are found in the epithelial lining, the mucosa of the stomach. So let me put mucosa at the top here. I'll say mucosal cells. Okay. And then the third type of cell is called a G cell. When we talk about the parietal cell is going to be making 2 things. It's going to be making and releasing hydrochloric acid. Hydrochloric acid is a very strong acid. It has a pH of about 1.5 to 3.5. So this is going to be very effective at denaturing pathogens that you've engulfed, and very important in helping to denature proteins, but potentially damaging to your tissue. So we're going to talk about, oh my goodness, how does the stomach deal with this very low pH? We will also see, and let's go ahead and say too, that the hydrochloric acid is important in activates pepsin. Okay. And then let's also say that the intrinsic cell, the parietal cell, makes intrinsic factor. And we're going to see that intrinsic factor is going to be important in helping us to absorb B12, which is a vitamin. And we're going to be absorbing that in our small intestine. We're going to come back and talk about that in just a little bit. So that B12 we've seen is important in hematopoiesis, which is the formation of red blood cells. And we can't get B12 without also making this intrinsic factor. So some individuals have anemia because they don't absorb B12 because they're not producing intrinsic factor. Okay. When we take a look at our chief cells, we're going to see that the chief cell is going to secrete pepsinogen, which is going to become pepsin in the presence of the hydrochloric acid. And pepsin, we'll see, is going to be important in helping to, I'm going to say, digest proteins. So if you remember when we talked about the mouth, we had salivary amylase, which was chemically digesting starches. We had lingual lipase, which was chemically digesting fats. And then we had the teeth and the tongue to mechanically digest proteins. We haven't seen any chemical digestion of the protein until we get here. So we're going to see the pepsin and the hydrochloric acid are important in digesting proteins. And then when we talk about the G cells, we'll see that they are important in releasing gastrin. And I'm going to do a whole talk on the hormones, and so I'm going to save our discussion of gastrin for that time. But what we can see is gastrin basically, I will say, stimulates the stomach here. And when we stimulate the stomach, think in terms of both the mechanical, the churning, mixing action, the segmentation movement, as well as the production of hydrochloric acid and pepsin. So these are all important chemicals. So we need to keep up with hydrochloric acid, pepsin, intrinsic factor, and gastrin. Okay, so make sure that we're paying attention with these. We're also going to see that mucosal cells are going to release somatostatin. And somatostatin is like continually released to inhibit gastrin. So basically, when we get into that cephalic phase, we're going to be stopping the release of somatostatin, which allows for the release of gastrin. So I'm going to say that again. When we're in that cephalic phase and we're getting ready, like, oh, I see food, I'm thinking about food, I'm smelling food, or I'm tasting food, and we want to increase our gastric activities, we're going to actually turn off the somatostatin that then turns on gastrin. And let me highlight somatostatin so we're keeping up with that one as well. So when we talk about gastric juice, we're expecting the gastric juice to have the hydrochloric acid, the pepsin, the intrinsic factor, and the gastrin, basically, right? And we're seeing that coming from gastric glands that are part of the mucosa. Now, the mucosa is epithelial tissue, and it's potentially being exposed to this like super strong acid, hydrochloric acid. So how is the stomach able to, deal with this? And what we'll see is that the stomach will have what's called a mucosal barrier. And the mucosal barrier is going to be a thick coating of bicarbonate rich. Remember, we talked about bicarbonate ion when we talked about transporting carbon dioxide gas, and we said it was a super important buffer in body fluids. So here we're seeing it sort of in action. So it's a thick coating of bicarbonate rich mucus that's covering the top of these cells. And then we also are going to have gap junctions between these cells to prevent like leakage so that we're not getting this hydrochloric acid like deeper into the tissue. So the reason the stomach is like okay at this super low pH is because of these special adaptations that this tissue has. The real significance of the hydrochloric acid is that, again, it's going to kill pathogens that you've either inhaled, remember that, or swallowed. And then it's also important for the activation of pepsinogen, and pepsin then is important in breaking down our proteins. So we're going to see that. When we are talking about the chemical digestion that's happening here. So when we talk about this chemical digestion, we're going to see, of course, pepsin and hydrochloric acid, which are going to break down protein. So we have pepsin and hydrochloric acid, important in breaking down proteins. And then we're going to see that your lingual lipase continues to digest lipids. So we're doing digestion of proteins in the stomach. We're doing digestion of lipids in the stomach, but I like to always describe the carbohydrate digestion as on hold, because that salivary amylase that you swallowed cannot function at this low pH. And so it's going to basically be denatured and ineffective. And so there's nothing new that's helping to digest the carbohydrate. So stomach, I consider proteins are being digested, lipids are being digested, and then the stomach is just, the carbohydrate, excuse me, is just on hold. The other thing just to talk about is what's called gastric emptying. And remember, we're seeing like 3 milliliter of chyme released at a time into the, here I'm just going to put small intestine because I have room for that. But, you know, we also can say, oh, it's going into the duodenum, which is the top layer. And so we'll see that the stomach is completely empty 2 to 4 hours after a meal. Again, depending on the amount of food that you're eating, but also the type of food that you're eating. So if we go from fastest to slowest, carbohydrates, meals rich in carbohydrates are digested first. Protein-rich meals are second, and lipid-rich meals take the longest to digest. So not only is there a difference based on quantity, but also type of food. All right, y'all. Up next, we're looking at the small intestine. So please stay tuned. As always, take care of yourselves and each other.